give this webinar. So I will stop sharing, and Raj, you can start sharing and give the presentation. Thank you so much for everyone.
All right. Thanks very much, Krishna, for the introduction. I'm very appreciative of you inviting me to this webinar. Today, I'm going to share some of my research I've been doing here in Louisiana for the past few years. So my title of my research is going to be Bioremediation of Explosive Contaminated Soil. So first half of my talk, I'm going to give a general overview of bioremediation, biodegradation process, and then I'm going to talk about the research that I've been doing here. So because of the wider audience, I just want to introduce the concept first. So if you already know most of the stuff I'm presenting, please bear with me. Okay. So first of all, let me introduce the topic of the biodegradation process and bioremediation methods. So biodegradation is basically use of biological treatment system to reduce the concentration of hazardous waste from contaminated site. So in bioremediation method, what we are doing is we're intentionally using the biodegradation process using microbial physiology and exploiting microorganisms to degrade these chemicals. So that's basically what bioremediation is. So let's talk about biodegradation itself. Let me see. So biodegradation is basically microorganism using the hazardous chemical as their organic carbon source to grow on them. So if you given favorable condition, all organic compounds will be degraded by microorganisms. And that's why we don't see a huge pile of waste we are throwing up in the environment. But in the chemical that we're producing, which are xenobiotic compound, these are a man-made compound and chemically synthesized, and microorganisms haven't seen this type of compound in their lifetime. So those compounds are a little harder to degrade because microorganisms don't have a necessary enzyme to degrade. They have to evolve new enzymes because these compounds are pretty new to them. So it will take long time for microorganism to degrade any xenobiotic compound that we put them in the environment. So some of the compounds become recalcitrant. Recalcitrant compounds are synthetic compounds that are very, very hard to degrade. As a result, they persist in the environment for a very, very long time. So the recalcitrant property of a compound may be because of the chemical structure itself. So the unusual substitution, like we put halogen in the organic molecule, like chlorine and other halogen. Sometimes there is unusual bonds, and also we have highly condensed aromatic rings, and the compound itself has excessive molecular weight, such as plastic. So these compounds are very, very hard to degrade. As a result, these compounds are going to persist for a very long time in the environment. So recalcitrant nature is not only chemical structure itself, but it's also biological as well, because microorganisms, as I said, haven't seen these chemicals before, so they don't synthesize the necessary enzymes. In order for a compound to go away, there has to be the enzyme to degrade them. And the enzyme has to be induced by the microorganism. So there's a whole mechanism of genetic sequence where they had to turn the gene on and produce the necessary protein, and then it started to degrade the compound. So the failure of making the necessary enzyme itself is a problem. Another problem is the microorganism has to transport the compound inside the cell most of the time, sometimes has to do outside the cell. So they don't have a suitable transporter on the cell membrane. We call that permeases. So lack of that for the particular compound also a problem for compound to persist. Sometimes the compound is not soluble. So when they are not soluble, then it is not bioavailable. So there is a bioavailability issue that keep the compound persist for a long time. And in some cases, the compound itself is very, very toxic. And so it naturally kills most of the good microorganisms in the contaminated site. So those are some of the biological reasons why chemicals that we throw in the environment persist for a long time. So the problem with some of this compound persists, it could potentially biomagnify in a food chain. So biomagnification is when these compounds are not degraded in the environment, in the food chain, in the whole ecosystem, the compound can be absorbed in the food chain and food web. As a result, the compound bioaccumulate and biomagnify when you go up in higher level of food chain. So biomagnification is defined as an increase in the concentration of a chemical as it pass through higher member of food chain. So for a chemical to biomagnify, it has to have two properties. The first one, the compound has to persist, obviously, for a long time for the chemical to be transported through food chain. The second one is the compound should be lipophilic. That means the compound has affinity to attach to lipids in a living organism. So when this compound is attached to lipid, it doesn't undergo normal metabolism. So it will bioaccumulate in lipids. And when a higher trophic chain, it magnifies. That's what the biomagnification is. So one of the classic example is the accumulation of DDT in eagles. So in this diagram, it shows that DDT at concentration in the water is really, really low. If you look at it, 0.003 parts per billion, very, very low and way lower than the EPA standard. So EPA is going to look into this at that time in the 60s, so it is not of a concern. But that compound had, one of those compounds that is recalcitrant and biomagnify, and you can see that DDT accumulate in the food chain, and in the zooplankton, the concentration increase, and then into small fish and large fish, and then the super predator like a bald eagle that has, higher doses of DDT, and it's almost wiped out the species. So eventually DDT has to be banned to recover the population. At one time in US, the body burden of the every person living in the US had a DDT concentration of 0.01 ppm. So they had DDT, almost everybody in their blood. So it is kind of, one of those serious situations that lead to banning DDT. But DDT use could be, beneficial, but at moderate use, not overuse. So now it's coming back in Africa. They're using it to control mosquito population for malaria control. So let's look at some of the terms when we talk about biodegradation. So the general definition of biodegradation is it's a biologically catalyzed reduction in complexity of chemicals. So it's a very broad definition of biodegradation. There are two processes. One is a slight modification of chemical by microorganisms to change the structure of the compound to reduce the toxicity sometimes. That term is called biotransformation. Anything that's slightly modified to a compound we call biotransformation. The ultimate goal in microbiology, environmental microbiology, is to achieve mineralization, which is the complete degradation of the compound, converting all the organic molecule to its inorganic constituents. All the carbon is converted to carbon dioxide, all the halogen or any other substituent is converted to inorganic constituent of the compound. So that's what we try to achieve, and that will make the compound harmless in the environment. So if you look at microorganism metabolism, most of the time microorganism, when you throw these new compounds, they don't necessarily evolve new pathways to degrade them. So they in their evolutionary process, they developed these 4 major metabolic pathways. Those are glycolysis, Krebs cycle, electron transport system, bed oxidation pathway. Any compound that they encounter, they try to modify the compound, put in any one of these metabolic pathway and shunt these chemicals into this already existing pathway. So the organism is not inventing a new pathways to degrade chemicals. So this is a general metabolic pathway microorganisms utilize, and they say, so if any new compound they encounter, they basically modify the structure of the compound, try to fit in any one of these pathways so they can degrade the compound. But obviously, in order to make this compound go away, there are a lot of factors that affect degradation. So you need to make sure the compound is amenable for degradation. That's why the bioengineers come to modify, create optimum condition for microorganism. So the degradation depends on both microbial factors as well physicochemical factors. So based on that, the compound will persist or go away in the environment. So some of the biological factors are, as I said before, genetic potential of microbes. The microorganism has the right genes to induce the right enzyme. Then bioavailability of chemicals. The chemical itself should be available. If it is not available, either it is absorbed into a clay layer, it's not be bioavailable. Microorganism is not going to spend a lot of extra energy to make it bioavailable. Then the contaminant structure itself is a problem, and then toxicity of the contaminant, and then environmental factors that dictate the degradation rate in the environment. So some of the common physical factors are the oxygen requirement, organic matter in the environment, and nitrogen, phosphorus, temperature, pH, salinity, water activity. All these factors dictate how fast a chemical disappears from the environment. So in another factor in bioremediation, biodegradation is when compound degrade, it's mostly by synergistic method. What I mean here is not one pure culture of bacteria can degrade the chemical. So there's cooperation of a lot of different species in the environment that degrade the compound. One group carries out first reaction of the compound, reduce the toxicity, and the second species take over and convert them to another metabolite and easily amenable, and that become a carbon source for third group. So most of the time, it is synergistic biodegradation. Very, very rare to see one single species of bacteria completely mineralizing the xenobiotic compound in the environment. So as I said, there are a lot of chemicals we throw them, like halogenated compound like TCE and PCE. The bacteria has an option of completely mineralizing them, which is obviously the good thing that we want. Sometimes they just modify and they biotransform, reduce their toxicity. For example, TCE can be dehalogenated to be dichloroethylene instead of trichloroethylene. So you slightly change the structure of the compound, which is biotransformation. So what microorganism is getting in biodegradation of these chemicals is ultimately they're extracting energy from the compound and utilizing this chemical as food source by oxidizing the organic chemicals that we are throwing at them. So there are 3 ways to do them. They biodegrade, they mineralize, they biotransform. So the techniques involved in bioremediation is broadly classified as natural attenuation and engineered bioremediation. So natural attenuation is basically monitoring. Almost everything is available to microorganism, conditions are right. So simply monitor the concentration in the environment. So it's most like a passive bioremediation. Engineered bioremediation is where we alter the condition to keep the optimum condition for microbes to degrade the chemicals. So the advantage of bioremediation is there is comparatively a little cheaper physical chemical method. There is no additional disposal cost because you are most of the time making the compound harmless by mineralizing the compound. Generally speaking, low maintenance, and the treatment system does not create an eyesore, and also is capable of impacting many source zones and decrease the cleanup time in many cases. So those are some of the advantages of bioremediation technique. So microorganism can use the chemical in 3 different ways: as a primary substrate in their metabolism. That is the ultimately easy way to get rid of a chemical, because the compound itself is serving as carbon source, so you don't have to add additional carbon source to grow bacteria. Secondary substrate is the compound is not in high concentration environment, but you want to get rid of the environment, so it's not serving as primary substrate, it's serving as secondary substrate. So it's going to take quite a long time to degrade such chemicals. And then the last one is co-metabolic substrate. It's an accidental transformation of a contaminant. In this case, you need to add an additional carbon source, easy carbon source, for bacteria to grow on. And then the bacteria growing on an easy carbon source, but on the same enzyme that is metabolizing the primary substrate, also degrading the xenobiotic compound. That is co-metabolic substrate. So some of the cartoon here, so you have the chemical itself, which is a toxic, and it could serve as carbon energy source. You need to have right electron acceptor. The aerobic condition is oxygen. Then we have various reducing condition from nitrate to sulfate reducing condition. So electron has to be accepted. So we need to provide electron acceptor in the environment. Then we have environmental condition like temperature, pH, and conductivity. Of course, you need to provide nutrients like nitrogen, phosphorus, and trace elements for bacteria to divide and multiply. So when the electron acceptor and electron donor are available, the bacteria are going to oxidize the compound and put the electron into electron acceptor and converting them to carbon dioxide and water. And all the organic constituent in the contaminant will become inorganic constituent, which is mineralizing the compound. So it can be done both aerobically and anaerobically. So most of the time, bioremediation achieved in aerobic condition. The oxygen is your terminal electron acceptor. So limiting factor is oxygen. So you need to provide oxygen to the contaminated site. Under anaerobic condition, the anaerobic bacteria can also degrade. So you need to provide either various electron acceptor like nitrate, iron, sulfate, or methanogenic condition, or fermentation condition also could be used under anaerobic condition to remove some of the compound. For example, if you have subsurface, you cannot economically deliver oxygen to create aerobic condition. You can kind of encourage anaerobic bacteria to make sure there is enough electron acceptor to encourage the growth of anaerobes to take care of it. In general, anaerobic degradation is a lot slower than aerobic because anaerobes take a long time to double. The doubling time is slow compared to aerobic condition. There's another group of bacteria that are called facultative bacteria. They can grow in the presence and in the absence of oxygen. So that's also advantageous to use in cleanup process. So here is aerobic process is basically oxidizing the compound. That's ideal. The compound itself is a carbon source, or you could use co-metabolic condition. You provide primary substrate for bacterial growth. In anaerobic condition, you have a variety of sources depending on electron acceptor available, like denitrification, nitrate is electron acceptor, manganese reduction, iron reduction, sulfate reduction. So you have various electron acceptor condition and methanogenesis, where CO2 itself is act as electron acceptor. So if you look at in the actual cleanup sites, the preferable electron acceptor is oxygen. When oxygen is depleted, the next level of bacteria available is nitrate reducing based on the redox potential of the environment. And then when nitrate is gone, then the next group of bacteria will take over this iron reducing condition, sulfate reducing condition, methanogenic condition. So you can see that in the graphic in subsurface. Cleanup, aerobic is the first one, and then it goes through denitrification, iron reduction, sulfate reduction, methanogen, based on the availability of electron acceptor by the compound to be completely oxidized. So here is some of the common compounds that are amenable to aerobic and anaerobic conditions. So if you look at, for example, acetone contaminant can be easily degraded both ways, aerobic bacteria as well as anaerobic bacteria. And if you look at some of the compounds, they are only amenable to aerobic, and some of them are only amenable to anaerobic condition. For example, PCE. PCE is really amenable to anaerobic condition at a highly biodegradable state as compared to aerobic condition. So there is, in the literature, people have done a lot of research to which are the compounds amenable to aerobic and anaerobic degradation process. So when you talk about bioremediation practice, so basically what you're doing is understand the physical, chemical, characteristic features of the site itself and contaminant itself, understand the bacterial metabolic pathway, what kind of organisms is in the environment, what kind of pathway are normally present, understand the environmental condition required to promote the, achieve the desirable population level of microorganism, and make sure all the gene expressions are already there, so the enzymes are produced. And then you engineer the condition to promote the growth of bacteria. So all of them are manipulated in bioremediation industry to increase the microbial population. So as I said, oxygen is commonly used. So oxygen is most of the time is a limiting factor in cleanup process. So all you need to do is provide oxygen, and most of the other factors are mostly available. So oxygen it could be the only thing that you engineer to provide in situ conditions. Oxygen can be, applied to the contaminated site in both ways, in dissolved form and also gaseous form. So in dissolved form, you can, use hydrogen peroxide that has oxygen in its structure. And also in gaseous form, bioventing process, you pump or draw air through the contaminated site, so the microorganism will get oxygen. Those are some of the common methods. Then the anaerobic condition, one of the common way bacteria degrade is dehalogenation reaction, where wherever there's chlorine, bacteria reduce them in, substitute the chlorine with hydrogen, which is the electron. So the compound itself is acting as electron acceptor in dehalogenation reaction. We have dehalorespiration, so you need to provide enough electrons. Sometimes in bacteria get electrons in nature from other organisms when they degrade compounds, but so you need to have enough electrons for organism to reduce the compound. That is dehalorespiration process. So here is an example of how perchloroethylene, which has 4 chlorine in its structure, bacteria could use that as electron acceptor. So if you look at this sequence of events, bacteria is substituting 1 chlorine and reducing them with a hydrogen. So PCE become TCE, and TCE become dichloroethylene and end up in vinyl chloride, and then ethane. So if this is reductive dechlorination, so the electron acceptor compound itself acts as electron acceptor, and you're substituting the chlorine and converting them to a harmless product. So in doing in this way, sometimes you activate the compound. So you need to understand whether you are mostly degrading the compound, reducing the toxicity. So when you do, for example, when you dechlorinate perchloroethylene to trichloroethylene, you're going to increase the toxicity. The toxicity of TCE is more than PCE. And same thing when you convert to dichloroethylene, it's 50 times more. So and the last one, vinyl chloride, is cancer causing. So you need to make sure you're not activating, make sure this, when this sequence of dechlorination happens, eventually this vinyl chloride also is converted. If the reaction stops here, then you created a monster situation. So you need to avoid. So you need to understand the whole concept of metabolic pathway, so you make sure that you're not degrading the compound. You're not activating the compound. And then co-metabolic process is basically you're creating accidental transformation. You're providing enough primary substrate to promote the growth of bacteria. There are a variety of electron donors you can provide. There is a list from various cheap sources of electron donors. Here is some of the list from acetate to butyrate and molasses, and a lot of different easy sources, milk protein, all that stuff. So you wanted to make sure that if everything is available, you can do natural attenuation, but it is not fast. But engineered bioremediation is the way to go. Most of the time, you can achieve cleanup in a faster time phase. So in bioremediation, what we're doing is you're creating this bioremediation triangle principle. So you have to have these all three corner of a triangle. You have to have your compound, you have the right microbe in place, you have all the nutrients. So you're trying to bring all these elements together. That's all you're doing in bioremediation. So microorganism will take care of cleaning up the contaminated site. So bringing pollutants, right environment with right conditions, physical factors, and right microorganism. All of them are bringing them together. So there is a variety of compound that can be easily biodegraded. There is a lot of list here. So you can do this in situ or ex situ. In situ is without excavation. Ex situ, you dig up and modify the site. There is a variety of methods: bioslurry reactor, biopile, land farming. These are all some of the commonly used bioremediation method. Land farming is another common technique commonly used for contaminated land contamination of oil spill, very commonly used. And then you can use subsurface contamination in situ method if it is groundwater contamination. So there is a variety of sources. The groundwater can get contaminated, from industrial spills to storage tank leaks and pipes and landfills. Groundwater get contaminated. So here is an example of BTEX contamination through underground storage tank. You have this plume of L-napple that get into the groundwater. Can be cleaned up in situ method by providing right nutrient. A lot of chlorinated compound comes through a variety of ways. Here is a dry cleaning facility that can contaminate groundwater using TCE, where they use in their facilities. The TCE is a DNAPL, which is dense non-aqueous phase liquid. It will go into the groundwater and settle at the bottom part because it's denser than water. And it become a permanent source of pollution because its solubility is limited. So there's a lot of problem with DNAPL and L-napple chemical because it is not soluble. Okay. So with that, I'm going to now specifically talk about my research. I kind of give you a broad overview of biodegradation, bioremediation processes. So we did research in this particular facility called Louisiana Army Ammunition Plant in Minden, Louisiana, which is northern part of the state. This site is contaminated with these three explosives. This is the Army ammunition site, has been used since the Second World War, and then they shut down production in the 60s, but the land is contaminated, close to 15,000 acres. The soil has a lot of unexploded ordnance, and then, in the form of TNT, RDX, and HMX, trinitrotoluene. RDX is this Royal Demolition Explosive or Rapid Detention Explosive, and then High Melting Explosive, which is HMX. These are the three major components that contaminate this site. And this is the site located in northern part of Louisiana. The town is called Minden. And as I said, over 15,000 acres since 1941. And all production is completely shut down now. It's only in caretaker status. The Army has to clean up the site before they can, hand over the land to the local use. So the most of the site is completely unused. It has to be cleaned up first. So we got involved, we got project from Army to look at whether we can do bioremediation technique. So we look at work plan, review the existing data, collect sample, did lab studies, and um, uh, recommended a method for cleanup. So our objective is to determine if are there any microorganisms in the soil that can degrade in the contaminated site. The very preliminary study to look at why these chemicals still there for more than 70, 80 years. Maybe there's the lack of microbes, right microbes. So we took the sample, analyzed first, characterized, and did the standard HPLC analysis. These are the various standard we ran. And then if you look at the soil extraction, you can see these three peaks that are the major compound, TNT, RDX, and HMX. So our concern is to try to remove these three compounds from the soil. So the concentration range vary from 4,000 to 10,000 milligram per kilogram of TNT, 800 to 1900 milligram per kilogram of RDX, 600 to 900 milligram per kilogram of HMX. So as I said, it's a big site. So we took randomly 50 different samples and come up with the concentration in this particular soil. So we did the traditional microbiology enrichment technique, isolation technique to look at what kind of bacteria are there, inoculate with the minimum mineral salt media with chemicals and look at bacteria that can grow. And then did isolate 22 pure cultures of bacteria. We screened them all. And then it's a tedious process to look one bacterial species at a time to understand what kind of biochemical processes involved in modifying the compound and ultimately degrading the compound. And just quickly, I'm not going to give you all data, just two bacteria that I'm going to give randomly the results. One is called Staph cohini, another is Bacillus levolacticus. So we expose these pure culture to various conditions. We provide TNT as sole carbon source, TNT as nitrogen source. Of course, we use a co-metabolic condition where we provided some EC carbon source. And we went through traditional method to analyze. And here is some data. When you provide to staff and bacillus, and here's the staff, here's the bacillus. When you provide TNT as sole carbon source, the bacteria is able to grow on TNT as sole carbon source. The reason they didn't degrade, the conditions were not right at the site. But so when you provide right condition with the right amount of nitrogen, phosphorus, and pH, they all grow. Here is a co-metabolic condition, carbon source, nitrogen source, and you can see the TNT concentration going down. And same thing with bacillus. So bacillus can use TNT as carbon source, nitrogen source. Of course, co-metabolic condition, you see better growth. And all these conditions, you see concentration of the chemical go away. But if you look at RDX, RDX cannot be used as sole carbon source. We can see no growth when RDX is used as carbon or nitrogen source. But when you use additional glucose as carbon source, they grow, they degrade at the same time. But when you use the chemical itself as carbon source, bacteria are unable to degrade them, this pure culture. Same thing with bacillus, okay? RDX cannot be used as sole carbon source. HMX is the same story. So these two isolate I'm giving randomly because we tested all 22 different isolate. And they all have the same features. They cannot use HMX and RDX as sole carbon source. They could use them under co-metabolic condition. You need to provide some EC carbon source. Then you can degrade the chemical. So we used, this is a small lab study I'm showing you now. We need to use this application part, how we can clean up in the real site. So we selected two methods, and that method we selected was soil slurry reactor and a land farming method. So we set up a small scale where we have two sets of reactor that use molasses as carbon source as a co-metabolic condition. And in the control, there was no co-metabolic condition, no, there is no molasses, okay? And you can see when we provided molasses as co-metabolic condition, the TNT concentration went down really fast. compared to no molasses condition. So molasses helped to remove TNT faster. Um, when you look at RDX are the same situation. It takes a longer time than TNT, but eventually the molasses receive reactor, you can see RDX concentration going down. When there is no molasses, when there is no co-substrate, RDX is still there in the soil slurry reactor. HMX is even persisted longer time, and then the molasses received reactor, the HMX concentration went down compared to a non-molasses reactor. Okay, so that's one set of treatment option we gave them. Soil slurry reactor will do the job, but it is expensive. You need to dig up the soil, put in a container, create a condition for better mass transfer. So alternative method was land farming, where we did the same set in a small scale. We had a molasses, non-molasses pan. We periodically add molasses in treatment, and we just aerated the soil by mimicking ploughing conditions. And same situation, when there is molasses in land farming, you saw this TNT go down compared to non-molasses. Same thing with RDX, molasses land farming removed it, and HMX as well. So molasses treatment removed them. So the summary of these techniques show that TNT, RDX, HMX can be removed in both methods, soil slurry reactor technique and land farming method. Of course, soil slurry reactor was faster and better, but it is expensive. So there is a trade-off between which method to use. Land farming could be used, but it's going to be taking a long time. So we need to find out what happened to these compounds. So we did a radiolabel study. So we did a radiolabel TNT and took a subsample and spiked them with radiolabel. And you can see TNT is mineralized when you have the molasses added one compared to soil slurry control. And you can see the various product, we converted 22% to carbon dioxide. Another 23% is used up as a cell biomass. Bacteria use them and make more body parts. And then various metabolites are also produced in fatty acids. And then the land farming did the same thing, okay? So we kind of not only modifying the compound, but we are also mineralizing them. Bacteria able to use this TNT as a carbon source and converting them to carbon dioxide, as well as use them to make more cells and more bacteria. And then we wanted to find out what happened under anaerobic conditions. That's aerobic condition I talked about. So under anaerobic condition, we used the sulfate reducing method. We provided sulfate as electron acceptor and promote the growth of sulfate reducing bacteria. And you can see when you have co-metabolic condition, the growth was better compared to when there is no carbon, only chemical is the contaminant is the only carbon available. So the growth was better when you have additional carbon source. Then we created a slurry condition exactly like I showed you, but this is all anaerobic. Only thing is sulfate 20 millimoles available as electron acceptor. And you can see in both cases, co-metabolic condition with the sugar went down faster. And sole carbon source also removed a little slower. And this is our abiotic control, where there is no bacteria in the system. We kill the bacteria. Just to show that we expanded this into a bigger reactor, it did the same story. TNT went down in sole carbon source. It took a long time in anaerobic condition. And we did the radiolabel study, and just a summary of what happened to the TNT. You can see under anaerobic condition, we did not mineralize to carbon dioxide. Most of the TNT is converted to acetic acid. You can see that because this group of bacteria doesn't have the terminal oxidation step. So TNT is converted to acetic acid, almost 50% of them. And we produce all the other metabolite, but most of them are acetic acid. And compared to killed control, I mean, this is pretty good. Our mass balance is really good. 96% of TNT radiolabel we recovered in different forms under anaerobic condition. So based on that, we constructed the metabolic pathway, what happened to this particular compound. So TNT is, of course, this trinitrotoluene is a toluene with 3 nitro group. That's the one that gives the explosive nature to the compound. The bacteria is initially reducing the nitro group to amino group. The NO2 in each position is reduced to amino group. And then amino group is broken down. The ring is broken and converted them to cyclohexone with the multiple steps. And then it went through beta oxidation pathway where 2 carbon at a time is chopped. And what we end up is a 2 carbon compound called acetyl-CoA, which goes to Krebs cycle, as I showed you at the beginning, which is one of the normal metabolic pathway in bacteria. And then 2 carbon at a time is removed. And ultimately you have acetic acid, which is a 2 carbon compound. So acetic acid is harmless. The other aerobic bacteria would use them as carbon. So in conclusion, I just want to say that we did a traditional enrichment technique and showed there are bacteria in there. Why the compound persists long time? Because there was not optimum conditions available for them to degrade. If you provide optimum condition, the bacteria can take care of either aerobically or anaerobically. So we recommended a couple of methods. The soil slurry reactor is faster, but land farming is cheaper. But the Army can decide what method to go to clean up. So I will stop here because of time. I'll take some question. I'll stop here.